Welcome to Chapter 7. This chapter is going to be all about metabolism. So we're going to be talking about enzymes and specifically energy, where we have different pathways that we have to go through. So this chapter is going to be kind of similar to Chapter 6 in that there's going to be a handful of definitions that you need to know. But this one is going to be pretty heavy on steps in pathways. So just make sure you review this chapter as much as possible in order to get comfortable with the material. You'll be happy you did. So metabolism in and of itself is going to be the sum of all chemical reactions within a living cell. And it's going to come in two flavors, catabolism and anabolism. So what these two forms of metabolism are essentially reverses of each other. So let's talk about it. Catabolism is going to be the breakdown of complex organics into simpler molecules. In essence, we are breaking down molecules into smaller parts in order to get energy. Okay? With anabolism, this one is going to build molecules from simpler ones. So literally the reverse of catabolism. And in order for anabolism to work, we need to use energy. So we have to use that energy to make those molecules. In order for metabolism to work with catabolism or anabolism, we are going to need to use enzymes. Remember that enzymes are going to be those proteins where we have some term and it ends in ASE. Remember, more often than not, that is probably an enzyme, but these guys are going to catalyze reactions. So let's talk about it. There are certain processes that could happen without an enzyme. It just won't be as fast or as reactive. So when we look at an enzyme, again, we are talking about a protein that is going to start some chemical reaction. Depending on what we're looking at, it just really kind of depends. We've looked at a few enzymes before. Again, remember that they usually end in ASE. So let's look at this graph here that shows us kind of what's happening in this reaction, either with an enzyme or without an enzyme. So it's going to be harder for a process to happen without an enzyme. The activation energy is going to be a higher requirement in order for that to happen. You can think of it like building a shed where without an enzyme, you can still build the shed with hammers and nails. But with an enzyme, it's like adding power tools. Yeah, you're still building the shed, but you have these tools that make it easier to get to that end result. So we have less of an activation energy required if we use an enzyme. It is making the process easier and sometimes shorter. It just depends. We are still going from that initial reactant and we are trying to get to a product. That is kind of the end all be all result. But enzymes make it, for lack of a better term, easier. Let's look at the structure of an enzyme. It's going to have three parts. The first is going to be the apoenzyme. This is the protein portion of the enzyme. It's going to be that larger portion, and it's going to be inactive. We have a coenzyme, sometimes called a cofactor. This is the non-protein component made of other ions or organic molecules. So we see that cofactor or coenzyme there in the middle. That is our activator. And then our last piece is going to be the holoenzyme. This is going to be both of those first two parts of the enzyme once they're together. They're going to fit together kind of like a puzzle piece. Remember, when it comes to proteins, it's going to have everything to do with shape. And the shape is going to refer to the function of whatever that product is, whatever that enzyme is supposed to do, whatever the job is that that enzyme is supposed to fulfill is going to be based on the shape that it is. So again, apoenzyme, coenzyme together make a holoenzyme. You can think of that first part of holo as being whole. That is the whole enzyme. Now, reiterating what I just said about the shape of the enzyme being important, it's very important because it's kind of like a very specific receptive puzzle piece for something called a substrate. The substrate is going to be whatever that pre-product is. It's going to be able to fit into the active site of our enzymes, and that is where essentially those puzzle pieces come together. Once the substrate is in the active site, the enzyme substrate complex is going to be created where you have the substrate and the enzyme essentially connected, where the enzyme is going to then perform whatever function it needs to do to transform the substrate into what we're going to call the product. So once that product has been formed, it changes the shape of the substrate and then is released from the active site. So it is no longer the substrate, it is now the product. It will not fit in the active site, and that's what causes the product to be released. For regulating enzyme action, we need to talk about these two types of enzymes. These are constitutive enzymes and regulated enzymes. So constitutive enzymes are always present and in constant amounts, regardless of how much substrate is present for those active sites. Whereas regulated enzymes are either turned on, which we call induced, or turned off, which is repressed, and it has everything to do with the amount of substrate that is present. So this is a way to regulate enzyme action. Another factor influencing enzyme reaction is going to again be the shape of the enzyme itself. Remember that they're proteins and shape relates to function forever and always. So if we change the shape of the enzyme, change the shape of the protein, it's going to cause the enzyme to not be able to function the way that it was, which sometimes we want this because we don't want the enzyme to constantly be causing a reaction. Sometimes you need it to stop. When a protein breaks down, we call this denaturing. This is usually due to temperature, pH imbalance, osmotic pressure, or other chemicals. And usually this is going to happen by accident. Okay, more ways that we influence enzyme activity are going to be types of inhibitors. A competitive inhibitor makes sense. I want you to imagine that it is competing with the substrate. Because remember, when the substrate is in the active site, that is when the enzyme is going to function the way that it needs to and processes the substrate into a product. However, with competitive inhibitors, it's going to bind with the active site so that the substrates cannot get into the active site and then be processed. A non-competitive or allosteric inhibitor is going to instead interact with the enzyme in a different way. It's going to interact at the allosteric site and bind there. What this does is it's going to cause a reaction in the enzyme so that the active site changes shape. And this is another way to make sure that enzymes cannot bind with substrates and then produce whatever product it is that they would produce. Another way to control enzyme function is going to be known as feedback inhibition. And this is a pretty neat way to control the products of enzymes. So what's going to happen is the accumulated product of an enzyme pathway is going to essentially act as a non-competitive inhibitor. And what this will do is at a later point in the enzyme's production of a product using substrates, that end product is going to bind to that initial starting point of the enzyme activity and act as that non-competitive inhibitor. This is going to happen when there is a buildup of product. So after we go through this metabolic pathway, through these enzymes, we produce enough product from the substrates that we are able to use that product to stop the production kind of at the beginning part of the line. So what this will usually do is conserve energy as well as maintain homeostasis or balance within the cell. So because nothing lasts forever and enzymes are no different, we have to have a way to create enzymes, which we're going to call enzyme synthesis. Remember, that is a way to make something. We are talking about making enzymes. But we have to be able to control this pathway and regulate it. So eventually, enzymes will wear out and break down, and this can cause problems in the cell. So in order to kind of help with this process, we have to be able to make those enzymes. So enzymes will be replaced. And this type of control is going to require the regulation of DNA and the protein synthesis machinery, which we're going to talk about on the next slide. So the image that you can see is going to be this process in order to make enzymes. Where we're starting with, that step one, is our DNA making RNA, and then RNA is going to make that protein. Remember, that's that central dogma of biology that we've been talking about kind of over and over. From there, that protein is going to be used for an enzyme. Now, that's fantastic, that synthesis of a protein into an enzyme, but it's not always something that we want to keep going on forever and ever. So enzyme repression is actually something that we need to incorporate, which you can kind of think of as like a physical roadblock to the DNA producing the enzyme. So it is going to stop the synthesis of an enzyme in this production pathway. Now, if enzyme repression is essentially a roadblock for DNA, and it does not allow the enzyme to be produced, then enzyme induction is going to be kind of the opposite of that process. It is going to occur when the substrates that are required for those enzymes specifically are present. So we're actually going to see a lab later on, and a little bit more in chapter 8 as well. But it is the P-Glow lab we will be doing later in the semester that is going to kind of help us understand this process a little bit better. But just put a pin in that and just know that we have enzyme repression and enzyme induction. Enzyme repression is going to repress the enzyme from being made, while enzyme induction is going to be the opposite, and it is going to happen when substrates are present. That leads us into metabolic pathways. These are going to be essentially a series of chain reactions in order to get to some product. So typically what we need is the product of one step to be used into the next step, and that's going to lead into that reaction. So it is chain reactions. Once that first part happens, then the next part can happen, and so on and so forth. There are oodles of pathways, and there's not always one clear-cut choice for the proper pathway to get to that end product. Sometimes It's not possible to go through what would be the best option for a pathway. And so there are other options that cells can take to get to the same result, the same end product. So let's go ahead and kind of start about the pathway, specifically the metabolic pathway of energy. Remember from chapter 6 that there are two ways or two options for an organism to bring energy into it, and that is through sunlight. Remember, phototrophs, they're going to use photosynthesis, or consuming other organic organisms or substances like carbs when we talk about chemotrophs. So, when we talk about this energy, we need to talk about the reactions that are going to happen to allow for those metabolic pathways to occur. So at the beginning of this chapter, we were talking about these two terms, catabolism and anabolism. That is going to be terms that refer to what happens to the substrate specifically. We also want to talk about the energy usage. So when we talk about what happens to substrate, I want you to think of catabolism and anabolism. So catabolism or catabolic meaning to break something down to create energy, while anabolism is building something and you have to use energy. There are going to be two other terms here that we need to talk about, and that is going to be a focus on the usage of energy, which these terms are going to sound very, very similar as far as their definition goes to catabolism and anabolism. And that is because they are essentially the same thing. But again, it's the difference about what happens to the substrate versus what is the energy usage. So we have exergonic and endergonic. Exergonic is going to be creating energy by breaking a molecule down. So I want you to think that catabolism and exergonic are very similar in what's happening. Endergonic is going to sound a lot like anabolism, where we use energy to build something. But again, I want you to remember that catabolic and anabolic is going to be what happens to the substrate. We are breaking something down or building something up. Exergonic and endergonic is going to be the energy use, creating energy or using energy. So up till this point, in a lot of our definitions, we have used terms referring to energy like create or use, but this is not really accurate in talking about energy. I want you to remember the law of thermodynamics, where energy cannot be created nor can it be destroyed. So when we talk about energy being made, quote unquote, made, in something like a catabolic or exergonic reaction, it is essentially just energy being extracted from a larger molecule, being broken down into a smaller molecule, and then moved into an energy storage molecule, which we're going to learn as ATP. But you probably already knew that ATP is that primary source of energy for most cells. I want you to think of charging your phone or putting gas in your car in this sense that you would add the energy, like you're not making it, but you are moving energy from the electricity in your house, I guess, using the charger into your phone, or you are adding energy into your gas tank. You are, in the sense that energy is the gasoline. In anabolic and endergonic reactions, this is going to be where energy is taken from an energy storage molecule like ATP and put into bonds to build a larger molecule. So again, it's kind of just the movement of energy and where it's going, where it's being stored, as opposed to creating or using it. So this movement of energy is going to be accomplished by moving something called electrons. And we're going to talk a lot more about electrons in the next lecture, but this is going to be through a series of reactions called redox reactions or oxidation reduction reactions, which again, we are going to talk about quite a bit more in the upcoming lecture. I am going to finish this first part of chapter 7 on this slide just so that there is a definition of oxidation and reduction in front of you, but we will start this next part of this chapter right at this slide. So I hope you enjoyed this first part. We are going to get into some specific pathways in the next part of this chapter, and I hope to see you there.